Welcome back, everyone. So, we've spent the majority of our time discussing eukaryotic gene control in terms of ways that we're controlling what genes are being transcribed, whether it's DNA methylation or histone acetylation or homeotic genes or transcription factors. And that kind of makes sense that this is a very important way that we can control gene expression by going at the start from making those genes being transcribed or not. But once you have mRNAs around, there's also still ways that our cells can control if those mRNAs are being expressed or not. Obvious things like how long the mRNAs are going to exist until they get broken down, or even how long a protein that's created is going to exist. But a couple of other ways that we're going to talk about involve RNA interference and microRNAs. So mRNAs can be controlled by this small class of RNA molecules called RNAi, RNA interference. So that is a way to have some small RNAs trigger the degradation of the mRNAs. Or they may inhibit the translation of the mRNAs, or they may inhibit the transcription of a gene encoding a particular mRNA. In this image, you have RNA, which is double-stranded, which is going to be a signal to your cell. RNA should not be double-stranded. My mRNA should be single-stranded. It must be an invader. Get rid of it. So we have this sequence of proteins that come and cleave the RNA into pieces so that that RNA is being destroyed because of that RNA interference. That's because double-stranded RNA is typically going to be something that your cell associates with being from a virus, because viruses do have double-stranded RNA. So when double-stranded RNA exists, that is going to cause your cell to knock down the expression of that gene. What happens first is that this protein dicer, this is a ribonuclease. That means it chops up RNA. It is going to find that double-stranded RNA and cleave it into smaller pieces. So instead of having your long mRNA, now it's been cleaved into smaller pieces. And these little pieces are called siRNA, for small interfering RNA or silencing RNA. The siRNA will combine with this group of proteins called RISC, R-A-S-C. That's an RNA-induced silencing complex. And one of the strands is going to be incorporated into the RISC and be degraded, this green strand here. And the remaining one will bind the siRNA to its target mRNA by a complementary base pairing. And if that's a close match, it is going to cause this mRNA to be degraded by Argonaute protein. If it is a close match, a perfect match, it's being degraded. And if it's partly a match, it'll inhibit the translation of that mRNA. So we have created a way for your cell to control if that mRNA is going to exist or be broken down more quickly. This RISC can also enter the nucleus and be guided to the DNA that's complementary to the siRNA. The siRISC can silence expression of sequences through DNA methylation. It can recruit proteins that methylate histones. And cells often even possess an RNA-dependent RNA polymerase that will amplify this response to control gene expression. Another type of controlling your RNAs are using microRNAs. So microRNAs are going to be produced by normal cellular genes in almost all eukaryotes, and they're going to silence the translation of the mRNAs. They will bind to and regulate the expression of genes that are different from the genes that make the miRNAs. So the miRNA is going to be initially transcribed into longer molecules called miRNAs, and fold into these hairpin loops. These miRNAs are going to be converted into pre-miRNAs through cleavage or breaking down through this enzyme dosha, which is a nuclear enzyme. So now you have a pre-miRNA. This gets exported to the cytoplasm, where dicer cleaves it to form an miRNA. The mRNA forms the mi-risk protein complex, which will inhibit the mRNAs with sequences that are complementary to the miRNA. If that mRNA is fully complementary, it'll be degraded. And if it's partially complementary, the translation of that sequence will be inhibited. miRNAs play especially important roles in embryonic development. One example we see is if we delete this miRNA called MIR12, that can cause defects in hearts of the embryonic mice that can lead to embryonic demise. And there are at least a dozen mRNAs that have been targeted for degradation by MIR12. Okay, I hope you have enjoyed this chapter. It's one of my favorites. And I will see you later. Bye.